大家好，那我们很高兴今天请到吴明如博士来帮我们做这个演讲。那在场我看到很多高手就不远千里而来这里来听这个演讲这个主题当然是非常有趣。那今天我们所长没有办法主持，我来主持一下。那简单的来介绍一下吴博士。吴博士是台湾长大的，当然就是慈济医学大学医学系毕业，然后当了个兵，然后短暂的在中研院当助理之后，2009到2015年在美国的Dartmouth University 拿了一个 PhD 这个是一个常春藤的名校，然后他在那边的是 Department of Microbiology and Immunology， 然后做的就已经是跟 cancer 跟 tumor 相关的 CAR-T 然后更复杂一点的这种 targeting 的系统。然后2015年到现在是2019，是在做 Postdoc 在 MIT， 那叫 Laboratory of Electronics 还是
Research-Laboratory of Electronics。
对，这名称看起来 Electronics 跟 Bio 好像扯不上什么关系，但是非常的 Bio， 非常的 Genomics 的一个部分。他的老板也非常有名Timothy Lu 也是台湾的后代。然后他4年其实在这里就发了一堆 paper， 然后拿了一堆 grant。那做的东西就是跟这个主题就是 synthetic biology、synthetic gene circuit 的这类的主题。那各位如果看他的 CV 就会有一些非常好的 papers， 然后他的一些 grant。然后再过几个礼拜或者几个月内，他就要到Dana-Farber Cancer Institute开始他自己的 laboratory。那他是从assistant professor 做起然后同时跟 Harvard 的Department of Immunology 是有一起的合聘的一个教职各位如果知道，在美国现在要从 Postdoc 要拿到教职，通常要很多年。他4年就在这样子的地方可以自己起，他的 lab 是非常的惊人的。那我相信今天这个主题可能是以 cancer， 然后可能以 synthetic gene circuit 的开始，但是可以应用的范围不只是 cancer 而已。那今天大家来，我相信会有非常好的收获。那我们欢迎吴博士。
Thank you very much for having me. It's my great pleasure to be here. And today I would like to share our work on synthetic gene circuit for cancer immunotherapy. This work is part of my overall motivations, which is to develop synthetic biology platform for basic research and for therapeutics. During PhD, I focus on cancer immunotherapy. I focus on overcoming one major challenges of cancer immunotherapy, which is a lack of ideal targetable tumor antigens. Therefore, I have developed several pan-tumor targeting strategies, including pan-tumor targeting CAR-T cells, BITEs, and nanoparticles. And all these strategies can target a variety of tumor types in vivo in mouse model. And all their designs are based on harnessing the pan-tumor targeting activity of natural killer cell activating receptors. For example, by harnessing natural killer cell activating receptor, the NAM1 and NKp30, I have developed over 15 different CARs. Each has a different tumor recognition domains or co-stimulation domains. And here are a few examples of them. In addition, I have also developed an scFv-based CAR, the Anti-B7H6 CAR. It can recognize the tumor-specific antigen of NKp30. So this system is even more specific than NKp30 receptor-based CAR. And here I show you this CAR. The anti-B7H6 CAR can effectively prolong mouse survival in the lymphoma model and also significantly decrease tumor burden in ovarian cancer model. In addition, I have also worked on a type of protein therapeutics called BITE. BITE is a fusion protein consists of anti-tumor domain fused with a T cell activation domain. And with BITE, one can elicit anti-tumor response without explicitly engineer the T cells. And here I show the BITE can also prolong mouse survival in lymphoma model. And because of this unique pan-tumor targeting capability and the therapeutic efficacies, my work and others from the lab has been acquired by an international biopharma, Celia, for further clinical translation. And Celia now is testing our approaches in several clinical trials to target 7 different cancer types. However, at that time, I noticed there are additional challenges in the field of cancer immunotherapy I wish to overcome, but I could not. And it is because I have limited toolboxes. So I started to explore, and I realized that the tools in the field of synthetic biology may help me build the solutions. So I started my journey as a synthetic biologist during Postdoc. Until now in Postdoc, I have developed 2 synthetic biology platforms. The first is synthetic gene circuit for cancer immunotherapy. It has the potential to overcome several current major challenges in cancer immunotherapy. And next is a high-throughput engineering and screening platform for identifying synthetic promoters with enhanced cell state specificity. And today I will discuss both parts. And 2 patents were filed according to this work. And this work has also been licensed to a startup company. So why do I pick synthetic biology for therapeutics? And it is because synthetic gene circuit has a lot of biomedical applications. Synthetic gene circuit, or in short, gene circuits, are a group of genetic components that can work together to perform a more complex decision-making processes. These circuits usually are encoded in virus or in plasmid and be delivered into patient body where they can use with patient systems. For therapeutic purposes, these circuits are generally built capable of sensing a disease state and responds with the therapeutic output to turn the disease state back to a healthy one. And what is cancer immunotherapy? It is the approach that harnesses patients' own immune system to attack tumors. It is one of the most promising cancer therapy now in clinic. For example, taking a particular anti-CD19 CAR to treat relapsed refractory childhood acute lymphocytic leukemia. For these patients, all of their tumors respond to any known therapies. But with this particular CAR T cells, 90% of them can reach complete remission, and the effect is also long-lasting. For example, this is Emily. She's the first child therapy, and now actually she is 6 years tumor-free. And because of this unique efficacy and long-lasting effect, CAR T cell therapy has been the first approved gene therapy in USA. And this is how the approved product looks like. A small bag of cells, but huge impact. So how is it done? First, T cells will isolate from patient bodies. And generally speaking, T cells can recognize and kill target cells, but T cells generally don't kill tumor cells. But if you program them, usually with retro or lentivirus, to express the chimeric antigen receptor, it is a fusion protein consists of tumor recognition domain fused with a T cell activation domain. And the tumor recognition domain will allow the T cell to recognize and bind to tumor cells. And upon binding, the T cell activation domain can directly activate the T cells. So following programming, this T cell will expand ex vivo and then reinfuse back into patient body, where they can search and kill tumor cells. It is very promising. However, several major challenges still limit this approach. For example, first, the lack of ideal targetable tumor antigen. Ideal antigen should only be expressed in tumor cells, but not on normal cells, because if antigens are expressed on normal cells, these CAR T cells can kill those normal cells as well, and this generally leads to very severe toxicity, and sometimes it is fatal. However, such antigens are rare, therefore limited the application of CAR T cell. Next, even if there is ideal targetable tumor antigen, upon reinfuse back this T cell into patient body, tumor-mediated immune suppression can immediately shut off T cell function. and render the therapy ineffective. What do I mean by tumor-mediated immune suppression? Tumor cells are known to use natural mechanism normally used to prevent autoimmunity or to shut off immune system or to shut off immune response when the immune system is no longer needed. For example, using its surface checkpoint proteins to interact with the checkpoint receptor on T cells and directly inactivate the cells, or by directly secreting immunosuppressive cytokines to inhibit the immune system as a whole. Approaches such as using checkpoint inhibitors such as anti-PD-1 antibodies to inhibit this interaction and revive T cells, or to directly administer immunostimulatory cytokines. While these approaches are very effective, the systemic administration of these agents generally cause very severe systemic toxicity. So immunotherapy has a lot of promise but suffers three major challenges. So we think, can we develop a strategy that can potentially overcome all these three challenges all at once? For example, to overcome the lack of ideal targetable tumor surface antigen challenge, how about a strategy that can interrogate intracellular tumor signatures? Because there are much more tumor signature inside the tumor cells rather than just focus on the tumor cell surface. And to overcome the tumor-mediated immune suppression, how about a strategy that can trigger a combination immunotherapy that can be designed to target multiple aspects of tumor-mediated immune suppression? And to overcome the severe toxicity, how about a strategy that can localize the therapy at tumor sites? An approach we come up with that can potentially overcome all these three challenges all at once is to develop a synthetic gene circuit that, upon delivery into patient body, it can first distinguish tumor cells from normal cells, and it will reprogram the tumor cells and force the tumor cell to produce immunostimulatory cytokines that is localized at tumor site. So what is the rationale of our approach? Our approach harnesses two very important anti-tumor resources. The first is the immune system. Just as each and every immunotherapy, it is very potent. But our approach also harnesses one largely ignored anti-tumor resources, which is the cancer cell themselves. Cancer cells are very powerful protein production factories, so it can actually create very high level of protein therapeutics. And furthermore, cancer cells are by nature localized at tumor sites, and it will allow the production of immunostimulators from within the tumor and localize the tumor. Why is that important? Because it will enhance the safety and efficacy. Our circuits encode with RNA component only. Unlike most other circuits, which is built with protein components, our circuit can minimize the non-specific immune response against protein component in normal cells. Secondly, our circuit can integrate multiple disease markers, so it can reach a more precise tumor identification. Therefore, it will only be activated in tumor cells. Therefore, it will lower the required effective dose of the therapy, because it will create high local concentration of the therapy surrounding the tumor rather than the high systemic concentration in the entire body. As an effect, it will significantly reduce the systemic toxicity. So, how is it done? First, circuits encode in virus and can infect both cells and normal cells alike. And when circuits get into the cells, it can sense and integrate the activity of two predefined transcription activity with a logical AND gate. So meaning only when both activities are high, our circuit cell cancerous. And this logical AND gate significantly increases the targeting precision. It decreases the false positive target rate from P when targeting a single marker to P squared when targeting with the AND gate. As an effect, it will be only activated in cancer cells. And it will trigger a production of combination of immunostimulators And this combination is very important. I will get to it a bit later. This combination can initiate a strong and sustainable anti-tumor immune response. So essentially, our circuit can directly engineer tumor cells and turn tumor cells against themselves. So how do we exactly design a circuit that can distinguish tumor cells from normal cells? Tumor cells generally exhibit multiple aberrant signal transduction pathways. And these pathways generally converge into transcription factor activities. So we design our circuit to sense such aberrant transcription factor activities. There are 3 modules for our circuit: Module 1, Module 2, and Module 3. Module 1 and 2 are the sensing modules. Module 3 is the output modules. And there are a lot of components in our circuit. But for now, we only need to focus on the GAD component. GAD is an abbreviation of a synthetic transcription factor, a fusion protein consisting of GAL4 DNA binding domain fused with VC16 transactivating domain. It is a very commonly used synthetic transcription factor for regulating gene expression. So, when transcription factor 1 is active, it can bind to the sensor of module 1 and drive the expression of module 1. And when we initially design module 1, we intentionally split the GAD coding sequences to 2 exons and insert a module within. So with this design, transcript can be further processed by splicing machinery and form a complete GAD coding sequence. And the microRNA will be the intron will be further processed by microRNA machinery and form a microRNA. And because we also put a cognate microRNA binding site at the end of GAD transcript, so this microRNA will go and bind to this cognate binding site and mediate the degradation of GAD transcript. So essentially, module 1 is a self-inhibition loop of GAD production. So when only transcription factor 1 is active, no protein is produced. But when transcription factor 2 is also active, it can bind to the sensor region of module 2 and drive the expression of module 2 transcript, which is microRNA sponge. It consists of multiple microRNA binding sites fused in tandem, so it can very effectively soak up the microRNA in the system. Therefore, GAD transcript not degraded, GAD protein produced. So essentially, the sensing module is an AND gate for 2 predefined cancer-specific transcription factor activities. Only when both activities are high, GAD protein is produced, and GAD will further bind to its cognate promoter and drive all the output expression we designed for the therapy. And to make our circuit work rapidly, we actually need to optimize each and every aspect of our circuit, including the 2 cancer sensors, and the intronic sequences, and the architecture and the sequence of microRNA sponge, and GAL4 promoters. But due to time limitation, I will not get into the details of circuit optimization, but I will discuss how we optimize the 2 cancer sensors in my second part of my talk. And we get 2 very good cancer sensors. One sense MYC activity, one sense E2F1 activity. So now we have built our circuit. We want to confirm that our circuit is indeed cancer-specific. So by using virus to introduce our circuit in ovarian cancer cells, which we design our circuit to target against, the red and the purple is using the single sensor to drive fluorescent protein output. The blue bar is using our circuit to drive a fluorescent protein as an output. And indeed, we observe our circuit trigger very high output in ovarian cancer cells. And we further introduce the same circuit into a panels of normal cells, including ovarian-related fibroblasts, ovarian-related endothelial cell, or normal colonic cells, or normal breast cells, or iPSCs. And we can see our circuit is basically minimal or not active in a panels of normal cells. So we know our circuit is tumor-specific. Next, we design a therapeutic output for our circuit to trigger immunotherapy. The output we design is called surface T-cell engagers, or abbreviated as STE. It's a fusion protein consists of an anti-CD3 antibody domain fused with a transmembrane protein. And our STE design can very effectively express on a variety of tumor cell surface. And upon expressed on tumor cell surface, STE can bind to the invariant portion of a T cell receptor and directly activate the T cells that it binds. And the T cell can directly kill the tumor cells that bind to it. And because of this interaction is independent of the antigen specificity of the T cell receptor. So STE can be seen as a universal antigen. So now we have a circuit that can trigger a therapeutic output. We want to validate whether using our circuit to trigger STE, can we get a tumor-specific immune response? So we introduce our circuit to express STE into both tumor cells and normal cells. And from now on, I color-coded the data from tumor cells in red, and the data from normal cells or the data from control cell states where circuit is not supposed to be active in black. So first, by using fluorescent staining, indeed, we observe only tumor cells express very high level STE on their surface, but not normal cells or control cell state. Next, we co-culture these cells with human T cells and use cytotoxicity assay to measure the percentage cell killed by T cells. And indeed, we observe T cells very efficiently kill tumor cells, but not normal cells or control cell states. And in addition to tumor killing, cytokine production is also very important. So we also measure the cytokine production in this culture supernatant. We measure because cytokine production can initiate a positive feedback loop of immune response. And we measure one very important cytokine, interferon gamma. And indeed, we observe only tumor cells trigger very high level interferon gamma production, but not normal cells or control cell states. And as you may know, combination therapy generally works better than single therapy. So we also design our circuit to trigger combination therapy. In addition to the STE I just mentioned, which designate expression cell to T cell mediating killing, we also design our circuit to secrete CCL21. It's a chemokine that is known to attract cells. Also IL-12. It's a cytokine. It's a very potent immunostimulator. And lastly, the anti-PD-1 antibody is a checkpoint inhibitor known to disrupt tumor-mediated immune suppression. And from now on, I will refer this combination as a SCIP combination. And this combination is designed to target multiple aspects of tumor-mediated immune suppression. So first, we confirm that indeed our circuit can trigger the production of the SCIP combination specifically from tumor cells, but not from normal cells or control cell states simultaneously. So now we know our circuit is tumor-specific and can trigger combination therapy. Next, we want to know the maximal therapeutic efficacy of our circuit. And first, we want to know that first we use lentivirus and introduce a circuit to a wild-type ovarian cancer cells and create a scenario that 100% of tumor cells express our circuit. And then we introduce these tumor cells into the peritoneal cavity of NSG mice. NSG mice lack an adaptive immune arm, so it allows us to implement human tumors, and it also allows us to implement human T cells to study a therapeutic efficacy in a clinical relevant setting. So how does it work? We measure tumor burden and mouse survival, and x-axis is the day post-tumor inoculation. And we can see the black line is a control group where tumor is without circuit. We can see tumor grow aggressively. How about the orange line? It's where tumor express STE only. We can observe a significant tumor reduction, but the mouse still have allowed tumor burdens. And this is actually very striking to us at that time, because when 100% of cells express STE, meaning 100% of cells can be attacked by 100% of T cells, we still cannot get complete tumor reduction. This really indicates how aggressive the tumor is. And this is also almost like the best case scenario with a CAR T-cell therapy. So how about skip combination? When secreting skip combination, we can see tumor burden decrease significantly and very early on, and the mouse remain tumor-free throughout the process. And this decreased tumor burden also translates to long-term mouse survival. And here's what the individual mouse look like. The controlled, multifocal, well-disseminated ovarian cancer cells. ST only, we get significant tumor reduction, but the mouse still have a lot of tumors. And here is a skip combination. Very early on tumor reduction, and mouse remains tumor-free throughout the process. And this really indicates combination therapy can work much better than single therapy. Next, as you may know, gene delivery can never reach 100%. Because if gene delivery can reach 100%, we might as well use in our circuit to treat apoptotic genes. But because gene delivery cannot reach 100%, yet our circuit trigger immune response, which can amplify. So we want to know whether when only a partial tumor delivered with our circuit, can we still observe a robust efficacy? So we simulate partial delivery. And we want to know the delivery efficiency in a very precise manner. To do that, we mix the previously engineered ovarian cancer cells containing our circuit disproportionately with wild-type ovarian cancer cells and use this mixture to establish a similar tumor models. And here's what we observe. The dotted line are the controls where tumor without circuit or with circuit but no T cells. We can see tumor grow aggressively. And the solid line are the therapeutic groups. For example, the blue line is when only 15% of tumor cells having our circuit and with T cells, we can observe significant tumor reduction, and this reduction also translates to long-term mouse survival. And this is really very exciting to us because 15% delivery in vivo in human is possible. Next, we want to understand the therapeutic setting in a clinical relevant delivery setting. Because generally it's not that feasible to take patient tumor out and engineer them and put it back to the patient. So this time instead, first we introduce a tumor and establish a full-blown ovarian cancer. Then we introduce our circuit encoded in lentivirus into peritoneal cavities. Then we introduce human T cells to observe the therapeutic efficacy. And here's what we observe. Control tumor grow aggressively, and with the skip combination, we observe very significant tumor reduction. And this reduction also translates to long-term mouse survival. And this is really very exciting because this is really a therapeutic relevant delivery setting. Lastly, because we design our circuit with high modularity, this is an ovarian cancer targeting circuit I just showed you. By simply switching the two sensors to another two sensors, we actually get a breast cancer targeting circuit that works very well. So for part one, the conclusion, synthetic cancer targeting gene circuit can potentially overcome several major limitations facing cancer immunotherapy, and it exhibits strong, robust therapeutic efficacy in vivo in mouse model, and it can be adapted to treat a variety of tumor types. So I would like to, as I mentioned previously, to make our circuit work robustly, I need to optimize each and every aspect of our circuit, and one major optimization is the two sensors. And this leads to the second part of my talk, which is a high throughput engineering and screening platform for identifying synthetic promoters with enhanced cell state specificity. Cell state specific promoters are very useful tools for biomedical applications because it enables one to secrete a therapeutic output or a reporter gene at a very distinct cellular conditions. And because of these capabilities, scientists are always trying to find cell state specific promoters for their own utilities. And one common way to identify it is to directly look into native human genomes, because promoters are generally directly upstream of coding sequences. It's relatively easy to locate them and clone and test for their specificity. And indeed, most cell state specific promoters in the literature are identified this way. However, they generally don't work very well. They generally have a good activity in the cell state of interest, but also have a significant activity in the cell state not of interest. And the activity difference is rarely higher than tenfold. And why is that? Because a native promoter can be bound by a lot of transcription factors. For example, take native H2A1 promoter as an example. It's a commonly used native promoter for targeting cancers. When we perform transcription factor binding analysis, each color boxes is a location for a transcription factor family to bind. We can see it can be bound by a lot of transcription factor families. And generally speaking, a lot of transcription factor in mammalian systems are activators. So binding roughly corresponds to activities. So for this promoter to be active in tumor cells, generally it's not a problem. For it to be not active in normal cells, you need the normal cells to not to express all these transcription factors, which is very unlikely. So we think, instead of using native cancer-specific promoter, why not design our own cancer-specific promoter, which is the sensor for our gene circuit, and we design with the same logic, just using one type of transcription factor binding site fused in tandem upstream of immunopromoter, and this constitutes our synthetic promoters or our cancer sensors. And when we perform a similar analysis, indeed, for example, this is one of the sensors I used in the first part of my talk. Indeed, we can see it's bound by much less transcription factor families. So does it really work more tumor specific? Here I show you two native cancer specific promoters. The red bar is their activity in normal cells, and the blue and green bar is their activity in two normal cell lines. We can see both promoters indeed have high activity in cancer cells, but also significant activity in normal cells as well, and the difference less than tenfold. And how about the synthetic promoter we built? We built panels of them. The one highlight is the two works for the first part of my talk. We can see both have very high activity in tumor cells and almost minimal or no activity in normal cells. And for this promoter, it can reach 255-fold activity difference between tumor and normal cells, and this can reach 66-fold. So indeed, it is much more tumor specific. And the way we build is to first go to TCGA and CCOA database to identify the transcription highly active in tumor cells. Next, we have another set of database and we know 50 of them. Two works very well. Although two works well out of 50, we consider this approach we have a relatively low success rate. And not to say indeed we have low success rate. To do this a lot of time, we don't even do that. But to do this whole pipeline, we need to have the transcriptome or the transcription regulation data from the cell state of interest. A lot of time, we don't even have that. And even we have that, we still suffer from low success rate. So we know we don't want to work with a system like this. We want to have a system where we can identify any cell state specific promoters without any knowledge of the cell state. And we want it to be robust and simple. And that's exactly how we develop. First, we construct a library of synthetic promoters constitute of almost all known human transcription factor binding sites. And we use it, each individual binding site fused together upstream of a minimal promoter and use it to regulate a fluorescent protein, a red one. And to identify cancer-specific promoter, for example, we introduce a library into both cancer cells and normal cells. And because each promoter may have different activity in both cell types, so the cell will become different level of redness. Then we first use fluorescent to sort the cells in different bins of redness. Then use next generation sequencing to read out the promoters in each individual bins. For example, the blue promoter is highly enriched in non-normal cells, meaning it's not active in normal cells. And yet it's highly enriched in very red cancer cells, meaning it's active in cancer cells. So it constitutes of cancer-specific promoters. So we logic, we can derive promoter with any specificity we want, and also with any specific activity level. So how does it work? X-axis is the promoter is obtained from a single round of screening. Y-axis, the upper part, is the raw activity of the promoters. And lower part, and for this screening, we try to identify promoters that can distinguish a breast cancer cell line from a normal breast cells. And for the lower part is the activity fold difference between cancer cells from normal cells. So we can see from left to right, we can identify a panel of promoters that are highly active in normal breast cells, and a panel of promoters that are highly active in breast cancer cells. And the activity difference can range 1,000-fold difference, whether tumor-specific or normal cell-specific. In addition, this screening platform also allows us to identify promoters that can distinguish cancer stem cells from non-stem cell counterparts. In addition, our system also enables us to find tissue-specific promoters. This is in vitro organoid culture derived from program differentiation of iPSCs. We can see each blob is a different tissue or different organoid. And we can identify promoters that is only active in the outer rim of the organoid, or the promoter is only active in the inner cell mass of the organoid. So part 2, the conclusion is, our SPEC platform allows us to identify synthetic promoters for potentially any cell state of interest without prior knowledge of the cell state. And these promoters exhibit very high cell state specificity. So for future research plan, I would like to discuss 2 aspects. One is gene circuit therapy, and one is CAR T cell therapy. But gene circuit therapy essentially engineers tumor cells, and CAR T cells directly engineer T cells. Both approaches will be complementary with each other and will be synergistic with current FDA-approved cancer immunotherapies. So for gene circuit therapy, I would like to discuss 2 aspects. The first is the next step to translate cancer targeting gene circuit to clinic. And then will be discussed about the future perspective of expanding gene circuit to treat diseases other than cancer. To translate our cancer targeting gene circuit to clinic, I would like to further explore new design and also optimize circuit performance in these 5 different aspects. For example, for the sensors, first I would need to identify sensors that can distinguish patient tumor samples from normal patient tissues. And to do so, I will directly perform SPECS library screening against primary patient samples. And primary tumor are known by its heterogeneity. So new SPECS library design and new sensor architecture that can be more resistant to tumor heterogeneity will also be developed. Next, for the computation unit, or the logic gate. The immediate next question to answer is the 2-input AND gate sufficient to precisely distinguish tumor cells from normal cells? And the answer is very likely context dependent. If a tumor is very similar to its surrounding normal tissue, it's very distinct. If the tumor is very distinct from its surrounding normal tissues, 2-input AND gate might be sufficient. But if it is very similar to surrounding normal tissues, we might need 3-input or even more input AND gate. And I thought about that. My circuit design actually can directly extend to multi-input and in theory limitless input AND gate in a single computation layer. Next, for the output module, I would like to identify the best output combination for clinical utility. And to do so, I will utilize both patient-derived xenograft and syngeneic mouse models. With both systems, I will have a more clear view of what's potential best output combination. Then I would also like to optimize delivery vehicles. Currently, our circuit encodes in multiple viruses, so the cell need to be infected by multiple viruses to get a complete circuit. So I would like to explore first HSV, herpes simplex virus. It has a very large DNA encoding capacity, so I can encode the whole circuit in a single virus. And I would also like to explore additional material-based delivery vehicles. Because material-based delivery vehicles generally can deliver a cargo containing all the circuit components all at once into a single cell. And lastly, I would like to implement safety switch for our system, allow physician to dose in a orthogonal small molecule to control the magnitude of our circuit output. For example, by switching the GAD into a small molecule inducible transcription factor, RTTH3, essentially we get an inducible system for our system. And to further expand gene circuit to target diseases other than cancer, I would like to further explore these three directions. First, the sensors. I would like to expand sensor repertoire. Currently, with the SPECS library, I have a sensor library that allows me to interrogate almost all known human transcription factor activities. But not all the diseases can be readily targeted or diagnosed at transcription factor activity levels. Some diseases, such as genetic disorders, might more readily diagnosed at DNA mutation levels, or neurodegenerative disorders might more readily diagnosed at protein concentration or protein aggregation levels. So new sensors that can sense DNA mutation and protein concentration will also be developed. And then for the output module, I would like to develop output module that allows me to have an enhanced temporal and dynamic control of output production. What I mean by that, currently output module triggers a skip combination all at once simultaneously. But that might not be the best strategy, because the best drug likely will be the right drug at the right time at the right place. So first, I would like to develop a system where physicians can use multiple small molecules to control the combination, the timing, and the combination of the output production. And furthermore, I will further encode this sequential drug production mode into the circuit itself, allow the circuit autonomously perform this drug production, sequential drug production. And lastly, sometimes even physicians don't know when is the best time to switch to another drug. It might best decided by interrogating the environmental cue of the treated tissues. So I would like to develop a system where the circuit can also dynamically sense the treated tissue environmental cue and decide when to switch to the next drug. Lastly, I would also like to expand the computation unit, mainly expand the logic gate, because it will enhance the versatility of disease targeting. Because now the diseases are relatively targetable as an AND gate, some diseases might more readily targeted by logic such as A or B parentheses and C or D parentheses and not E. So additional logic gate will be very useful for this aspect. For example, there are a few examples of additional logic gate. Here are a few examples of them. And all this design can be implemented with a very similar logic of microRNA and sponge interaction. And all those designs can be accomplished at a single computation layer, meaning the circuit is relatively compact and the computation is faster and also more robust. Those are very important features for potential successful clinical translations. And for CAR T-cell therapy, I would like to bring forth synthetic biology back into CAR T-cell engineering. And the pan-tumor targeting system I developed and the logic gate and the SPEC platform will directly allow me to do this. For example, first, I would like to develop sense and response gene circuit to improve the CAR T efficacy against solid tumors. And I would also like to develop a tumor recognition AND gate for allow CAR T cell to be more tumor specific and target more tumor types. And lastly, I would also like to develop a safety switch for CAR T cell to prevent severe systemic toxicity. So to summarize, first, synthetic gene circuit potentially can overcome several major challenges facing cancer immunotherapy and exhibit strong therapeutic efficacy in vivo in mouse model. and it can be adapted to treat a variety of tumor types. Second, synthetic promoter library, the SPEC platform, allows us to identify synthetic promoters, allows us to identify cell state specific promoters for potentially any cell state of interest and without any prior knowledge of cell state. And this promoter exhibit very high cell state specificity. And lastly, synthetic biology provides powerful tools for basic research and for therapeutic applications. So with that, I would like to thank everybody from the Lu Lab, especially team, for being a very supportive mentor, and also all the team members that work on this project, and also our collaborator from Phil Sharp Lab, Yuval Tabak Lab, and from MGH, Sam Rapkin Lab, and our funding resources. And with that, I would like to take any questions.
非常谢谢吴博士很精彩的演讲。那我相信，如果开放问题会问不完，所以我们个别的，如果未来谈合作的问题，我们可以等一下大合照完再来问。然后我们先问一些 general 的问题，有没有什么 general 问题要提出来的？这次
Very interesting talk. I have two questions. The first one is on your part one.
Yes.
That you use the CMYC and E2F for the ovarian circuit, which is fine. That most of the ovarian cancer cells are CMYC driven. But how you also mentioned some tumor heterogeneity.
Yes.
That we know there are different subtypes. Not only ovarian and breast and all the solid tumor. How do you solve that? Are we going to introduce, like what you say, different combinations of circuits, and how do you control the ratio, or do you need to control the combination?
Yes, this is a very the first question. So this is a very important key point to address, especially for clinical translation, the tumor heterogeneity. I mean, exactly what's the proportion of tumor heterogeneity we need to address in clinic, we don't know yet. Yeah, for example, as a mouse model, when 15% of tumors are having a circuit, essentially you can see as almost like the heterogeneity causing only 15% of tumor response to the circuit, we still get a complete reduction. But for really specific clinical translation, indeed, the SPEC screening, we will need to screen for sensors that can, ideally some sensor can cover a broader spectrum of ovarian cancers. Now, generally active in a variety of ovarian cancer subtypes, or we can perform a personalized approach for screening against a personalized ovarian cancers. That's one major way to sort of really overcome and try to target as much as possible. But the first step will be try to identify the sensor that are generally active in most of ovarian cancer subsets.
Yeah, that will work, but because the experiment is the 50% is you still is at the background of one cell line type,
yes.
Right? So probably one solution to have a model for that is to mix different.
That's a very good one. Yes, exactly.
And then the second question is more technical about your part two. How do you determine your promoter? Do you look at their histone states to actually look at where they are the true promoters?
Oh yeah, we first perform the screening. So then once the screening hit identified from next generation sequencing, we clone each individual and really introduce to the cell state to see it's really active. So basically if a promoter you put into the cells and the cell light up red, we know it's because of promoters.
Do you think enhancers will work?
Enhancer, actually our design of synthetic promoter actually to some extent works very like enhancer. Enhancer often having multiple transcription factor or activator bindings. So it's almost like synthetic enhancers. But if we directly use enhancers, likely because there are multiple types of binding sites for a variety of activators, it's possible it will not be less specific, probably cannot reach a 1,000-fold. And so the way we go for, more a native enhancer, I guess. Synthetically, it's almost like a synthetic enhancer in our case.
好，那我刚才跟吴博士聊了1小时，我确定他听得懂中文啦，所以各位用中文、用英文问或用日文大概都有，都可以听得懂。还有没有再来的问题？
中文，就是你那个治疗还是 depends on local injection， 就是有点像 oncolytic virus 那种感觉。
这个 model 其实因为这个 ovarian cancer 大部分就是 intraperitoneal dissemination， 所以最直接包括 clinical relevant 其实就是往 IP injection 就是说事实上如果 systemic diseases， 一个是可以直接做 inject in systemically， 或者直接针对几个 primary tumor 去做 inject， 就是说 does not need to restrict it to local injection。但这个 model 就是 proof concept， 它当然就整个 peritoneal cavity 有很多 normal peritoneal cells， 理论上它也有机会是避开。
临床上比较大问题，病人通常是有 multiple tumor metastasis， 所以如果它的 effect 是在 local， 因为您强调是在 local immunity activate。
对，就
你们有看到，比如说 IV 注射或是 local 注射，但是其他的地方总有 response， 所以像什么 abscopal effect 有看到这种情况。
在这个 model 我们并没有特别去看它，就是说因为是发 ovarian cancer， 然后是我们第一个 model， 像我们有一些 breast cancer 的 work， 所以还没有做到 in vivo， 可是但像这种 obstacle effect 都非常重要的观察。当然就是其他的免疫疗法的确 variety of 免疫 immunotherapy 是有看到这种 obstacle effect 是没有错。
有意思。
Hi, I would like to ask in English, just it's easier sometimes. So you mentioned tumor heterogeneity. I am a little curious. Can you elaborate more on how diverse the promoter or gene circuit landscape would look like? Because it will definitely affect like which kind of combination you want to do.
Yes, the real answer is really only when we do it we know that. But I would say in general now from literature, depends on how you define heterogeneity. But when you do clustering, sometimes you get 4 or 5 major clusters, or those clonal evolution, you see a few major clones adding up a few 4 or 5 major clones or major subclones. So ideally, the more we can address, the better. So one way to address it on our system level is you really screen for promoters and identify, let's say, 3 or 4 different promoters that can cover a variety of cancer populations. That's actually meaning introduce multiple sensors. And the reason I say introduce multiple sensors is actually the sensor design is multiple in tandem. It can be split as binding site A, B, C, those combinations. So even we address almost like OR gate for transcription factor activity. So that's actually the first step to address these particular challenges. To build a sensor that can sense 2 or 3 common transcription factor related to tumor growth, and then another sensor 2 or 3 sensing a transcription factor related to like triggering additional control of tumor like apoptosis or trigger additional tumor signature. Yeah. So the key is really using multiple sensors yet build it compact.
Okay.
And I guess one thing probably I didn't mention strong enough is one important thing of immunotherapy is although tumors are all very heterogeneous, but they all share a common feature, which is somatic mutations. So meaning our circuit, even without deliver 100%, as long as we believe, as long as it can trigger a strong and sustainable immune response to a certain extent, the native immune system will take over to kill those tumors having mutation but not directly targeted by our circuit. So we need to really also, this circuit we believe it will also be able to harness the native immune response in the body.
Yes, I was going to ask, is there, I just suggest maybe there are kind of tumors that are more genetically related to certain genes, like in renal cell carcinoma, it's largely VHL mutated, and then maybe in there you have a more, like maybe other circuits that are very dominant, and then you might be able to test that.
That's a very good point. Yeah,
So any other question?
我用中文就好了。我想问的是那个这种疗法的安全性的问题，就是刚好没有提到说到底在老鼠有些被治愈，有些有没有有些在 treatment 过程中是被打挂的，因为我们是皮肤科医师，然后这种人造，至少 immunotherapy 出来，我们常被叫去看这种人造的各式各样有些致命的一些副作用。
本身这个实验这几个老鼠模型，这些13个 mouse model run 100% partial delivery or lentivirus， 我们并没有看到 obvious toxicity from mice。当然就是说免疫疗法本身的确，当 overexpression 有可能造成一个 positive amplification loop， 所以这也是为什么我们特别强调我们用 gene circuit localized immune therapy just at tumor site。尤其这个系统怎么说？当肿瘤全被杀光的时候，这是一个 self-sustainable 系统，就 self-sustainable yet self-limited。当肿瘤都不见的时候，理论上 immune response 也停了。对，的确回到就没有，我们并没有看到 obvious toxicity。
所以短期的 acute adverse effect 没有看到，但是有没有养久一点，说不定过半年有一些 autoimmune disease 就跑出来。
对，这其实之后一定要观察。不过因为这个模型它是 NHG mice， 所以它其实大概120天甚至更早就会有比较严重的 graft versus host disease， 所以其实一般是没办法做 long term observation。当然就是像之后 syngeneic mouse model 那些老鼠也就是几百天，那的确就可以看，包括如果以 lentivirus 可以看有没有什么 insertional mutagenesis。对，这之后也都一定要很注意去观察。
很高兴今天吴博士能够来这里给这个非常精彩的演讲。那这个会有 video 存下来，会放在我们的网页上面。那各位今天没有听得太清楚的，或者是要把他的 paper 念一念再来听的都可以。那我们来照个相喽，大家。